Hi guys, welcome to Seeker. Today I'm going to talk about the research which has been done on DNA repair mechanisms in cancer development and therapy. Actually, DNA damage has been long recognized as causal factor for cancer development. When erroneous DNA repair leads to mutations or chromosomal aberrations affecting oncogenes and tumor suppressor genes, cells undergo malignant transformation resulting in cancerous growth. Genetic defects can predispose to cancer. Mutations in distinct DNA repair systems elevate the susceptibility to various cancer types. However, DNA damage not only comprises a root cause for cancer development but also continues to provide an important avenue for chemo and radiotherapy. Since the beginning of cancer therapy, genotoxic agents that trigger DNA damage checkpoints have been applied to halt the growth and trigger the apoptotic demise of cancer cells. Here in this research, it has been provided an overview about the involvement of DNA repair systems in cancer prevention and the classes of genotoxins that are commonly used for the treatment of cancer. A better understanding of the roles and interactions of the highly complex DNA repair machineries will lead to important improvements in cancer therapy. Alright, here let's talk about the transmission of genetic information now. Of course, living organisms have the crucial task to preserve their genome and faithfully transmit it across generations. Transmission of genetic information is constantly in a selective balance between the maintenance of genetic stability versus elimination of mutational change and loss of evolutionary potential. The DNA molecule is under the continuous attack of a multitude of endogenous and exogenous genotoxic insults, and it has been estimated that every cell experiences up to 105 spontaneous or induced DNA lesions per day. Endogenous damage can result from DNA-based lesions like hydrolysis, deamination, depurination and depyrimidation, and alkylation or oxidation by intracellular free radical oxygen species that can occur as byproducts of mitochondrial respiration. Mutations can also arise during normal cellular metabolism, for instance by erroneous incorporation of deoxyribonucleotides during replication. Environmental sources of damage can be physical, for example ultraviolet light, ionizing radiation, and thermal disruption, or chemical, for example chemotherapeutic drugs, industrial chemicals, and cigarette smoke, and their effects varies from the formation of cyclobutane pyrimidine dimers and pyrimidine 6,4-pyrimidone photoproducts following UV exposure to the introduction of single and double DNA strand breaks upon IR treatment, or to inter and intrastrand DNA crosslinks, which results from various chemotherapeutic drugs. DNA damage occurs on a daily basis by endogenous and exogenous sources. Distinct DNA repair systems recognize and remove lesions. When the damage remains unrepaired, DNA damage checkpoints can halt the cell cycle or induce cellular senescence or apoptosis. Erroneous repair or replicative bypass of lesions can result in mutations and chromosomal aberrations. When mutations affect tumor suppressor genes or oncogenes, cell might transform into cancer cells. Therefore, DNA repair is essential for preventing tumor development. However, once a cancer has developed, DNA damage can be exploited to reduce cancerous growth and evoke apoptotic demise of cancer cells. Thus, chemo and radiotherapies are still today, over 60 years after having been first introduced into tumor therapy, important strategies to fight cancer. Given the central role of genome instability in triggering and treating cancer, it is likely that genotoxic treatments will remain an important avenue of cancer therapy. Also, the better understanding of DNA repair systems will allow therapies that specifically target selected repair pathways. It will be of particular importance to gain a deeper understanding how the various DNA repair systems interact with each other in the context of cellular homeostasis and DNA metabolism in order to optimize targeted approaches to cancer therapy. So thank you so much for watching this video, and don't forget to subscribe and please comment your ideas on comment section.